So first I want to thank the organizers. Michael, thank you so much for the invitation. I also want to thank you, Elle, for being so incredibly helpful with all the arrangements to come here. I want to thank the organizers for organizing an event that's pressing us to ask big questions. So often in bioethics, we're asking really small, lowercase letter questions. And here when I look at the agenda and I look at the speakers, we're asking capital questions, and it's very exciting to me. I also want to criticize the organizers for not inviting some more lowbrow speakers. It would be great to be able to blend in here easier, but anyway, I'm going to do my best. Okay, good. So let's get started. Oftentimes it's said that drug development is nasty, brutish, and long. It's nasty and brutish because we have to put a lot of compounds into this black box of development in order to get a small number of compounds out that look safe and effective. Also, it's nasty and brutish in that we have to put a lot of money into this black box, something like $1 billion in order to develop a single effective drug. So not only is it nasty and brutish, but it's also long. It takes somewhere around 8.3 years to go from a drug in phase 1 clinical trials through to a licensed product. And it's this sort of model, this understanding of drug development, that often leads to a kind of justificatory logic for the following two claims. The first is that the high pricing for drugs is justified. After all, if we didn't price drugs generously, no firms would ever enter into such a risky and lengthy endeavor to develop a new drug. So we have to keep high drug prices in order to attract private actors to invest in this very, very risky and difficult undertaking. And the other sort of claim that often follows on the tails of the model that I just described is this idea that we got to do better, that somehow we need to have higher success rates, that we should, instead of be putting 100 drugs into development to get 10, it should be more like 50, or that we should be able to develop drugs cheaper. I'm not going to fully disagree. I think both of those claims are problematic, and I can talk about why I think they're problematic. But I'm not really going to center my discussion about those claims. What I want to propose is that we often misconstrue what is morally at stake with clinical development when we assume that is the model of what is happening when we develop new drugs. And the crux of what I want to argue today is that, at least morally speaking, if we want to think about drug development, we need to have a different model of what exactly is going on, a model that's much more sensitive to the moral stakes of the enterprise. Number one, the main output of this enterprise is not really drugs, it's evidence. And I'm going to describe the kind of evidence that we're getting out of this process. So if we really want a good drug development system, it's going to be one that's going to generate the kind of evidence we need in order to run equitable healthcare systems. The other big criticism I have of this model that I want to emphasize is that really what the main input is into this enterprise is not so much a lot of compounds. It's not really that many dollars per se. It's human and non-human animal welfare. We put a huge amount of welfare into this black box in order to develop this evidence, and that if we want to have a morally sensitive system, we really want to have a good exchange rate, essentially, between the welfare we're putting in and the evidence we're getting out. And that way, we can think of what I'm going to talk about as a kind of moral economy. We get some exchange of welfare for evidence about how to use drugs and run healthcare systems equitably. My talk's going to divide into 3 different sections. First, I want to describe my model, my more morally sensitive model of what exactly clinical translation is, or drug development. I want to talk about some of the inefficiencies or some of the ways that our exchange rate here could be on unfavorable terms and could be perhaps improved. And then I want to close by talking about some policies we might think about that might move this towards a more favorable exchange rate in this economy. So let's start with the model of clinical translation. Probably everyone who in this room is familiar with this kind of a map of what happens when we develop a drug. Typically, drugs are developed in a laboratory, testing in non-human animals. They move into clinical development through phase 1, phase 2, and phase 3 testing. With phase 3 studies, you have definitive establishment of a drug being safe and effective. You cross that threshold into regulatory approval. Now a drug company can market that drug, and there may be subsequent studies, phase 4 studies, that are sort of tweaking or elaborating on certain aspects of the drug. So, you know, this is a fine way of understanding clinical translation. It's a map, and like all maps, there are certain aspects of the enterprise that it really highlights. other aspects backgrounds that are sort of invisible. And I would argue that there's a lot of moral stakes of clinical translation that model backgrounds. And like all maps, it's sort of something that kind of tells us how to get from point A to point B relatively easily. But if it's not highlighting what morally matters, it's not going to really help us get to where we want to be morally when we undertake this process of drug development. So a couple key features I've already alluded to about this map of drug development that I want to emphasize is that it really understands mainly the input into this process as being about compounds or drugs or dollars, and it understands the output primarily about in terms of drugs. So let me try to make the case that really, if we want to think about this black box well and in a morally sensitive way, we really need to first think about the welfare we're feeding into the system. In order to develop the kind of evidence about how to use drugs, we have to do certain forms of science. In particular, we need to do research using non-human animals and human beings, entities that have moral status, entities that are potentially going to give up welfare in order for us to develop the evidence we need about whether or not a drug is worth reimbursing, whether it's worth being used at the bedside. I'm not going to talk about non-human animals. I'm going to stick to human animals here in this talk. And I want to describe some of the welfare sacrifices that patients need to make when they go into clinical trials. So any patient going into a clinical trial is going to have to endure the side effects of a new drug that may not be effective. They're going to have to endure the psychological uncertainty of having a treatment assignment determined randomly. No patient opts or chooses to be randomized, so they're going to have to endure that uncertainty. They may endure research procedures, biopsies, venipuncture, and whatnot. Their lifestyle may be regimented in order to adhere to the clinical trial protocol. Et cetera, et cetera. Caregivers also are implicated. If you're in an Alzheimer's trial or an ALS trial, your partner is going to have to be the one that's going to transport you to the clinic and to help you adhere to the clinical trial protocol. I would argue that all of this is a form of work. It's a form of unpaid labor. We don't really adequately recognize it as unpaid labor, but it's unpaid labor much in the same way that household labor and volunteer labor and slavery and all sorts of other things. I'm not saying this is slavery, but I'm just saying there are all sorts of different kinds of labor that is uncompensated, and this is a form of labor that's uncompensated. Now, you might be puzzled, why is this uncompensated? Ellen Brandt actually alluded to this a little bit earlier. We sort of strike a bargain when we do drug trials that patients are in a state of what Ellen Brandt called precarity, and that if you go into this trial, there's a possibility of benefit. That claim is not entirely wrong. But it's also not entirely right. If you think about this logically, if you are doing a study that is not in clinical equipoise, and it's a randomized control trial, you're either going to be randomized to something that's inferior to your standard of care, or a drug that might be competitive with standard of care. So there's probably going to be a net welfare loss for patients in a study that's not in clinical equipoise. If you have a study that's in clinical equipoise, you're going to be randomized to a standard of care, so your quality of care is probably going to be pretty similar to what it would be if you didn't go into the study. Or you're going to be randomized to a drug for which science has not yet provided decisive evidence about its superiority. It might, in fact, turn out to be worse. That's why we're doing the clinical trial. So on the whole, although this is a nuanced position, and I can elaborate on it more, on the whole, I think it's fairly safe to say that if there is a benefit that patients get from going into trials, it's likely to be very nominal on the whole. So my group has actually tried to quantify the amount of human welfare that goes into developing a drug. We all know how many dollars it takes. We know how many compounds it takes. How much human welfare does it actually take? We did this in a very crude way, but I want to underscore that it's important to measure and to think about this. So what we did is we looked at a large sample of drugs, some of which got approved and some of which didn't get approved. And we simply counted the number of patients who had to participate in clinical trials to get a single drug approved. And in neurology, about 67,000 patients had to participate in clinical trials of neurological drugs in order to get 5 drugs approved. In cancer, about 100, my glasses aren't on, so I'm going to say, I think it says 159,000, had to participate in cancer trials in order to get 13 drugs approved. In both these cases, by the way, it turns out the number needed to treat is about 13,000 patients. In other words, 13,000 patients in a state of precarity, as Alan Brandt would say, have to be fed into this black box in order to develop a single effective drug in both neurology and cancer. And we can actually ratchet up the standards of what we mean, what this ratio is, if we look, for example, only at those drugs that deliver moderate to high value using standards of the American Society of Clinical Oncology. It turns out that we need about 40,000 patients per drug. So now if we scale this up, if we think that there are every year something like 20 different cancer drugs that are approved, et cetera, we're now talking about basically moderate-sized cities, probably cities maybe about the size of Jerusalem, of patients, that have to participate in this enterprise in order to, and have to give up some of their welfare in order to enable us to develop the evidence that we want to run our healthcare systems. Let me turn to talking about the drugs. So why do I think drugs are not really the output of drug development? Well, the main reason is because drugs don't do anything. They just sit in a bottle or in an IV bag. Drugs don't do anything unless physicians know exactly how to unlock their power in order to affect a therapeutic benefit. And so really this task is to evolve the evidence that we need in order to convert a drug that is born toxic, born dangerous, born a form of mayhem, it's born a poison, and to transform it into something that has therapeutic value. Now that process unfolds over basically 2 or 3 different steps. I'm going to describe them, but there's 3 forms of evidence we really need to get out of that process, and that we want our translation systems to be attuned to developing this in order to run healthcare systems properly. The first is that we need to know the materials, practices, and beliefs that we need to combine with a compound in order to unlock its therapeutic value. I call that an intervention ensemble. And this happens in early phase clinical development, phase 1, phase 2. So here's the basic idea, cell therapy for ALS. The cells don't do anything, they're just gonna sit in an IV bag, unless you know what quantity of cells to deliver, how to isolate those cells, what kind of immunosuppression regime you need to give patients while you deliver the cells, when in the course of ALS to deliver the drugs, et cetera. That's what we mean by intervention ensemble. So the first task of clinical development is to identify the necessary and sufficient components of an intervention ensemble. And I want to say another word about that. Many of the different components of the intervention ensemble are scalar quantities. They take on values, numeric values, like take something like dose, such that there's going to be an optimal dose, and there's going to be some boundary, a lower or upper boundary, below which the drug is no longer safe and effective, or above which it's no longer safe and effective. And so a key task in building that intervention ensemble is identifying the neighborhood of that window for the lower and upper boundary. Now, that's not just a sort of intellectual observation. That's crucial for healthcare systems to run effectively and efficiently. Let me illustrate this with a hypothetical stroke drug. Let's imagine we have a neuroprotective stroke drug. We know that if patients come into the hospital 4 hours after an ischemic event, if we deliver the drug, it's protective, great, patients have less disability down the road. Well, some patients are going to show up at the hospital 6 hours after their ischemic event, or 8 hours, or 12 hours. At that point, is it still worthwhile to deliver this very expensive drug? Are the side effects worth putting up with in order to deliver this drug? We can't answer that question unless we have defined the outer boundary on that timing variable for delivering this stroke drug. So establishing the necessary and sufficient components and the boundaries around them within intervention ensemble is the first piece of evidence that we really need in order to run effective healthcare systems, equitable ones. The second form of evidence is probably the most familiar to us, and that's the idea of taking an intervention ensemble, putting it to a rigorous evaluation, preferably with a standard of care, and showing that you can achieve a benefit that is exceeding the standard of care. That's what happens typically in late phase. Now, oftentimes the story of what kind of evidence we generate in clinical translation ends here, but there's a third component that I think is just as crucial, which is that in order to use these drugs, we also have to understand something about the theory of the drug, the pharmacological theory, but also the pathophysiological theory. You're developing a new drug, your drug may show effectiveness, but there may be some things that you learn about how the drug interacts with the patient that leads you to tweak that drug, that you acquire some sort of theoretical knowledge and achieve even better efficacy down the road with a modified drug. In addition to that, every time you run a clinical trial, you are generating information on various you're getting feedback on various pathophysiological and pharmacological theories. Now, these theories are crucial to applying the drug because, as we know, physicians don't treat populations, they treat individual patients. So every physician is always going to be in the position of having to interpolate evidence from a population to an individual patient or extrapolate by applying a drug to a population that does not meet the eligibility criteria of the clinical trial. How do physicians do that interpolation and extrapolation process? They do it all the time at the bedside. That is ultimately going to be held together by mechanistic knowledge, by theoretical knowledge, much of which is generated in this process of trialing. So if we're going to have an effective clinical translation enterprise, not only are we going to have to be mindful of the welfare that we're putting into this process, but also making sure we're capturing high quality evidence of those three different components that are crucial to know how to use a drug. And again, in that way, I think of what we're talking about with clinical translation as a moral economy, because there's some exchange that we're doing here between welfare and our output. Let me turn to talking about some of the inefficiencies, some of the various ways that our exchange rate is on less favorable terms than it could be. Now, I could stay here all day and elaborate on all the different things one sees when you pick up medical journals that, to me, have an unfavorable exchange rate. I'm not going to go through all of these. I'm just going to kind of describe a couple. For example, 2-to-1 randomization, something you often see in clinical trials, late phase clinical trials. That sounds pretty benign. What could be wrong with that? You've got clinical equipoise. Well, what's wrong with it is that it takes about 12% more patients in order to achieve the same state of knowledge about your primary endpoint in order for a clinical trial like that. And I've seen lots of studies that use 2-to-1 randomization. I've rarely seen a protocol that explains why they're using it in a way that's particularly convincing. That's an example, one of many different examples of routine practices that occur in biomedical research that I think don't really do justice to the importance to maximize the exchange rate between the welfare we put into this system and the output. The other point is that there probably are zones or areas of research that probably have more favorable and less favorable exchange rates. There probably are certain clinical hypotheses or intervention approaches that are more wasteful, not really sort of good value. I think all the work that we've done on vitamin D is probably a good example of extremely wasteful research. I always joke that that vitamin D is so ineffective in anything that you're not even getting false positives with vitamin D studies. But I want to talk about one particular zone where I think we have an unfavorable exchange rate, which is post-approval research or label extension research. Oftentimes after a drug gets approved, drug companies will often then try to test it against other indications. And at least my research would suggest that the exchange rate, the sort of the value we get for society, plummets after a drug gets approved. I want to illustrate this with a study that our team did looking at the drug imatinib. Now, imatinib, a drug that was developed in the late 90s for chronic myelogenous leukemia, is sort of the poster child for the perfect, efficient, personalized medicine drug development approach. Hopefully many of you have heard of imatinib. First in human clinical trial, 70% response rate for a disease that was at that time untreatable or refractory. That's almost unheard of, a 70% response rate. The very first indication it was tested in went on to get regulatory approval in a mere 3 years. That sounds like a great exchange rate, and it is. But if we want to talk about the efficiency of translating imatinib, we can't just talk about the first indication in which it's tested. We have to look at the whole body of research testing imatinib against various malignancies. And it turns out that if you look at all the indications, this is a diagram that is describing all the indications on the vertical axis for which imatinib was tested. And on the horizontal axis, it's the year, and each node represents a clinical trial. Those green arrows are indicating the cancer indications for which imatinib went on to get a regulatory approval. You see that early on in drug development, there's a cluster of indications for which there's FDA approval, and later on, there's a lot of indications for which there's a lot of testing, and there aren't regulatory approval. So imatinib is tested ultimately, at least at the time we did this analysis, in 42 different indications and 41 discrete drug combinations. And 14 of these go on to get regulatory approval. Now, if you look, sort of zoom in on various parts of the map that I showed you, you see basically a tale of two cities, to come back to this moral economy metaphor, perhaps. On the one hand, it's the best of times when you look at all the trajectories or indications that are put into development before regulatory approval. So before regulatory approval, 80% of the indications tested used enrichment design, which means that patients were tested to see if they had a molecular marker that matched imatinib activity. They were only included in the trial if they had that molecular marker. So there was really good, careful use, sort of pathophysiological theory in those trials. 2/3 of those indications go on to get regulatory approval, so it turned out those were good hypotheses to bet on, and 45% of those trials are positive on their primary endpoint. This looks really, really great in terms of a good exchange rate. But let's look at what happens after regulatory approval. 46%, so less than half of these trials, are using enrichment designs. In other words, they're not matching the drug to a molecular characteristic of the patient population. Only 6% of these go on to regulatory approval, and only 13% are positive on their primary endpoint. And you see this not just with imatinib, we've done studies on all sorts of other cancer drugs. And the net upshot is, it seems funny and sort of paradoxical to say this, but generally speaking, if you're a patient, the best place to be in terms of your welfare being transformed into clinically useful knowledge is in the initial tests of the drug, not the late ones when we actually know something about the drugs. So I would suggest that with late, post-approval research, we probably are in a zone where there are less favorable terms for exchange. Let me talk about another sort of inefficiency or another area where we have an unfavorable exchange rate, and this is the phenomenon of clinical agnosticism. This also is in the context of post-approval research. So normally we have phase 1, phase 2, phase 3 testing. We can think of phase 1 and phase 2 as exploratory, building that intervention ensemble, phase 3 as confirmatory studies. And the idea is that if you get a positive signal in your phase 2 study, you have good reason now, a good moral justification, and scientific one, for now running a phase 3 clinical trial. Oftentimes, however, there is a long gap between the positive signal in a phase 2 trial and initiation of that confirmatory testing. And if your drug is already approved, and this is an off-label indication, physicians can write prescriptions on the basis of very weak phase 2-like evidence for that, and patients will pay out of pocket, or to some extent, healthcare systems will reimburse, even though this hasn't been promptly put into rigorous testing. Let me illustrate this with the drug pregabalin. This is a drug that's approved for post-herpetic neuralgia as well as a bunch of other conditions. This is another one of these diagrams where you see all the indications for which it's tested, and you see the year. Each node is a clinical trial. The green, again, indicates the indications for which the drug went on to get regulatory approval. And the question we wanted to ask is, for how many indications were there cases where the phase 1 and phase 2 led to a positive signal, meaning that you would now want to rigorously evaluate this in a randomized trial, but there was a long, or what we called a prolonged period of agnosticism, another way to put this is clinical equipoise, where there was never a phase 3 trial launched to confirm the clinical activity from those small provisional phase 2 clinical trials. And the crux of what we found is that there were 27 cases where there was positive signal obtained, but that for 63% of these, there was prolonged agnosticism. In other words, for 63% of these indications for which early phase studies suggested there might be efficacy, they were never actually put into randomized, rigorous, large randomized trials to confirm that within 5 years. Again, drug companies don't really have much of an incentive to do this after a regulatory approval. In fact, if physicians will prescribe drugs based on the possibility they might work, based on agnosticism, as opposed to conviction, then actually it's a risky proposition to invest in a clinical trial to potentially disconfirm that your drug is going to be promising. And so as a result, in a lot of cases, these positive early phase signals will get picked up in clinical practice guidelines as recommendations. So this is kind of an interesting, observation, because here you have a case where I would argue that the clinical translation process, you might say it's generating some information. I would say it's generating more uncertainty. It's generating provisional knowledge that a drug might or might not work. We know that most drugs that work in phase 2 do not actually show efficacy in phase 3, so a lot of these are probably likely to be ineffective. So I think this is a system that is essentially putting more medical uncertainty into our healthcare system, not exactly what we want in a moral economy. I want to talk briefly about another sort of set of phenomena, what I'm calling agnostology of intervention ensembles, why it is that we don't know certain things in medicine as we would like to know. I'll go through these really quickly for the sake of time. Let me just skip that one. Let's talk about drug deprescription. Our systems of drug development are really, really good about telling us when a patient is indicated for a drug and getting them started on a drug. Our system is really, really bad about identifying which patients are suitable for being withdrawn from drugs and how to withdraw those drugs. I'll give you an example. Relapse remitting MS, it's known that patients, as they get older, their immune systems senesce, meaning that it is probable that many MS patients no longer need to take powerful, potentially risky disease-modifying therapies when they're in their 60s, 50s, 60s, and 70s. And yet there's very, very little research testing, using randomized trials, testing whether or not patients who have MS in their 60s do better when their disease-modifying therapies are withdrawn. And you can go to any number of different areas, osteoporosis, epilepsy and whatnot, and find this problem that we get patients on drugs and we don't really know how to get them off very well, something that would be crucial for running a fair and equitable healthcare system. Another is drug dosing. Oftentimes drugs are approved on the maximum tolerated dose. There's all sorts of commercial reasons and scientific reasons why that's the case. But it turns out that a lot of drugs are probably going to be just as effective, if not more effective, at lower dose. Many drugs are ultimately prescribed at lower doses than they are approved. Again, we don't have really a good post-approval research system that is testing optimality of dose and sort of figuring out whether we can deprescribe or lower the dose of that drug. Drug sequencing is another problem. Patients typically start a drug. They may ultimately fail that drug and need to go on a different drug. Let's go back to the case of MS. The paradigm for treating MS has always been to start with the least invasive therapeutic approach and to escalate up to the more aggressive disease-modifying therapies. There's an emerging paradigm, the inverted pyramid paradigm, where you start with the really hard-hitting disease-modifying therapies, risky, but it's hopefully stops MS in its tracks. And then if the patients stop responding to that, then you de-escalate. There's only now, I mean, people have been talking about this for 10, 15 years. It's only now that we're starting the first randomized trials testing which of these paradigms. So these are examples of major questions, huge import, that our system of clinical translation is not geared to resolve. Essentially, we are left with a huge amount of uncertainty, and that is a product. It's not a byproduct, it's actually a product of the way that we organize our research system. Let me turn to talking about some policies that we might use to sort of up or improve the exchange rate. So at first I want to talk about is regulatory gatekeeping, this idea that we have regulatory authorities that decide whether to approve a drug for commercialization or not. I would see this, I'm not a policy expert and I'm not an economist, so take this with a grain of salt, but I would see this as one of the most important mechanisms we have in order to maximize the exchange rate or make it at least most favorable in terms of the welfare we feed into the system and the quality and type of evidence we get out of that system. So prior to approval, companies can't sell that product, so they have very strong incentives to run rigorous clinical trials in the smallest number of patients possible, as quickly as possible to get across that threshold of approval. After approval, drug companies no longer have as many regulatory obligations. They have very little regulatory obligations to continue to conduct certain kinds of research. And as a result, oftentimes drug companies will pursue that research more slower than policymakers would like if the FDA has asked for post-approval research. So what that means is if we start approving drugs earlier in the process, we're basically shifting who is bearing the medical uncertainty. We're redistributing the medical uncertainty. We still have the same amount of medical uncertainty at phase 2 approval, right? Whether we're approving at that line or not approving and requiring a phase 2. So the question is, how are we distributing that medical uncertainty? And I would say that not only are we making the exchange rate less favorable, but also the way we're distributing that medical uncertainty is less equitable. I want to illustrate this in 3 different ways. First, as I mentioned, pre-market, drug companies want to do this as cheaply as possible, so they'll use the smallest populations as possible to evaluate their interventions. That sounds good. You're minimizing the amount of welfare that you're feeding into this unproven drug in order to acquire a certain amount of evidence. Whereas post-approval, this is now on the market, lots of patients are exposed. Yeah, we can use real-world evidence kind of to get this information, but that's a lot of patient exposure and a lot of data cleaning that you're going to have to do in order to get a clear signal. So that's one way in which we're redistributing. Another way is we're distributing from a privileged population to an underprivileged. We know that clinical trials, thanks to the Belmont report and all sorts of other changes, today clinical trials in patients with disease tend to occur in relatively privileged populations. If you look at eligibility criteria, they tend to be the healthiest of the sick. And generally speaking, you see more affluent white populations that are going into pre-licensed clinical trials. If you now shift regulatory approval earlier in the process so that patients are getting exposure to that uncertainty in the context of care, it means you're no longer primarily burdening a privileged population in order to acquire that evidence. There's the financial aspect. Before approval, private actors are the ones that are paying for the financial costs of evolving that evidence. If you now shift this to regulatory approval earlier, it means that our public healthcare systems or patients themselves paying out of pocket are now the ones that are bearing the costs of evaluating that evidence. And the last point I want to make is that if you have exposure within a clinical trial, there's an imperfect system of informed consent, but it is a system of informed consent whereby patients are told that they are part of an evidence-generating process and that there's risk and uncertainty for participating in that study. If you now have early approval and most of the exposure to that drug is in a care setting, there really aren't proper or rigorous consent processes to explain that uncertainty and that knowledge-creating enterprise. So broadly speaking, I tend to be someone who looks very skeptically at proposals to move approval earlier in the process because I think it leads to a less equitable moral economy and also one with a less favorable exchange rate. There's lots that research ethics committees could do. I see I have about 32 seconds, so I'm going to breeze through these super quickly. By the way, I love that clock up there. So I think research ethics committees, we talked about, is there too much regulation or not enough? I think research ethics committees, I know like I've got a reputation for saying things like this, but like I think they're incredibly acquiescent about some very, very flawed designs that we use in clinical trials. I think 2-to-1 randomization will be a perfect example where I think IRBs actually should ask the question of why you want 12% more patients to participate in that trial to get an answer. They should be seeking to minimize risk. There may be good reasons for 2-to-1 randomization, but it should be explained and investigators should be able to provide a clear answer for that. IRBs also don't require researchers to conduct a systematic review or even a landscape analysis to look at other drug indication, to look at other trials testing the exact same drug in the same indication that preceded it. And as a result, with surprising frequency, there are trials that go forward that are testing a drug in an indication for which there are several previous conclusive trials that already addressed that question. So I think IRBs could do much more in terms of asking for a more scientific justification. One thing I want to pick up on from Alan Brand's remark is that we're in a very different research economy today than we were in the 1978 when Belmont was written. Number one, at least in the United States, your salary support and your promotion is entirely dependent on your being able to get funding, get those R01s, to support your research. So there's very strong incentives to do research no matter what. As well, there's this whole other private sector for which there's a huge amount financially at stake in getting doctors to have certain kinds of beliefs. You want them to believe your drug is effective. And in that way, the research enterprise is very much shaped by these actors. And I think IRBs really need to sort of catch up with the reality that, evidence generation is not just a purely benign undertaking. It has enormous stakes. I'll skip this. Let me just close with a conclusion. So the net upshot, what I want you to take away, if you remember this talk 10 seconds from now, your working memory still has a little bit left in it. This is all I want you to take away, that we can think of drug development not so much as this kind of linear process where, we put a lot of drugs, and money in order to get an approved drug. That's a great model if you're a pharmaceutical company. God bless them, please continue doing what you're doing, no problem. But I think if we want to be morally sensitive to what's going on, we really need to think about this as an enterprise that uses a very large amount of human welfare, a large amount of non-human welfare, mostly uncompensated. We can talk about that a little bit more. It's mostly unpaid labor. And we need to be attentive to making sure that we steward the gift of that unpaid labor effectively when we think about how to regulate and oversee this process. And what we're getting out of the process is not a bunch of pills that are going to sit in a bottle or in a warehouse. It's pills with the evidence about how to unlock their therapeutic value. And so it's crucial that we design our systems so that we're capturing and disseminating and taking up the information and evidence that we generate from that arduous process of clinical development. I'll close by just thanking some of the people in my research team. These are the top ones are some of the trainees that did some of the work in some of the data that I presented. And the lower two are my close collaborators and friends, Alex London and Deborah Zarin. They are not to blame for any of the lapses in what I presented. I'll be clear about that. I'm truly blessed to have such incredibly generous collaborators and students. And of course, I'm blessed to have the support from CIHR in Canada. Thank you.